Catherine Williams. So this goes kind of a little bit with the clinical trial question. So Dr. Graylow, you were talking about how the I-SPY 2 trial was so creative and it was, thinking outside the box, and had set it up so that they could really quickly determine if treatments were working and kind of throw out the bad ones and move on with the good ones. And I'm just curious why that's not more common. Seems like that would be a really good way to determine which treatments were working and, improve overall survival for metastatic breast cancer patients. Thank you.
Right. This is a big effort that lots of sites are participating in, and I don't know that we need 10 different versions of this. I think that would do us all better to all join together and participate in the same big project. But I hear what you're saying. It was a new concept. It took some creativity. It took having multiple pharmaceutical companies come to the table and agree to let their drugs be mixed in and out and all. I think there are some metastatic trials where we've also seen this, the ongoing match trial with the NCI, that I think we have 24 drugs or something available now in different pathways. I'm the SWOG executive officer, not just for breast, but for lung. I know nothing about the treatment of lung cancer, but I know how to do clinical trials and get clinical trials approved, so I can be the executive officer. There's a lung map trial that's very creative. I really like that, and it's fun for me to see it and see if I can move something like that into the breast arena. But I watched the development of that trial where we had patient advocates and the National Cancer Institute and the Foundation for the National Cancer Institute, which is where donor money comes in. We had the FDA, and we had, at the first meetings, 5 different pharmaceutical companies all at the table with SWOG and the clinical trial list, trying to all do the right thing and get this big, complicated trial going. And it was thrilling to see how well everybody worked together. And in I-SPY 2, we had some very strong people who were able to get this all through. But it wasn't easy. It's not always easy to get that. But we have precedent for it now. And I think we will have more of these kinds of trials because we have precedent for it. We've had all kinds of people sit down at the table working toward the same goal of trying to break down barriers and get promising drugs to patients who are actively dying, now, soon.
And there's a very early breast version of the MATCH trial getting started by the Alliance Foundation with Monica Bertinoli and Otto Metzger and several others, where as it'll be built little by little with trials coming in as we develop drugs, targets that can, for which a drug will work. And, as several of the speakers have pointed out, there are not that many mutations in breast cancer that we know match very well to a drug, but there are a few. And there are some other findings that are not necessarily mutations, but other cellular phenotypes that can help us match. So it'll be a more organically slow-growing one compared to the lung one, which was sort of built, the whole house was built first and then run.
I think it's also important to remember that a lot of these studies, they're exciting and they're meant to jumpstart things, but they're not definitive trials. They can give you a first look, and as you heard already today, the veliparib drugs, that PARP inhibitor, there was a signal from I-SPY that it might be active, but the definitive trial, I have to say, was pretty disappointing. That, at least whether it's definitive, the veliparib carbo combination. So it's good to be looking at more creative, more rapid ways to take all these new drugs that are out there and at least get some signal about which ones should go forward. But they can't substitute for the definitive trials where you really test them and say, okay, is it really better or not? It turns out we're not that good at predicting always what should happen. And probably those endocrine studies from today were the most sobering example of that, where for a long time it seemed like more was better, longer was better, and then there was this data that maybe that wasn't true. So I'm not at all arguing against I-SPY 2 or the MATCH or any of these. We have to find faster ways to bring these drugs along, as you suggest, but they won't be the last thing, or we will not have responsibly really proven that they work.
Can I just ask something about I-SPY 2 that maybe you and the advocacy committee can help us with a little bit? And that's the concept of getting chemotherapy before surgery, which is how I Spy 2 was designed. So I just wonder, for women who had operable breast cancer, how many of you were offered that, and you got chemotherapy? How many were offered chemotherapy before surgery? Yeah, see, it doesn't happen very much. And some of that is just a limitation in trying to, the advocacy piece that I would like, and we're all medical oncologists here, so we all think the same thing. The most important part of breast cancer management is the medical therapy. The surgeons keep presenting that no matter what they do, it doesn't make a bit of difference, and that actually, so I have if my surgical colleague were here, he would admit the same thing. So the idea that getting the medical therapy right in the adjuvant or neoadjuvant setting is the focus we want to, that I personally talk to a lot of women about in participation on I Spy 2. There's a concept that once a diagnosis is made, the breast needs to be taken care of, and actually neoadjuvant chemotherapy does that, and it doesn't have to be surgery. So I think that for us, in terms of accrual to I Spy 2, that is a barrier that hopefully we're getting around. But I think, again, the idea that you can help us with that when you have friends or colleagues who ask you for your opinion about what to do, that will help a lot.